Oh, y'all got quiet, so it's a good time to get started, I guess, even though we're like 30 seconds early, maybe. So exam Friday, the mean in here was about 74. So right now I'm like 98% sure I'm not going to curve that with such a high mean. Sorry, let me turn this down a little bit. That's a little bit better. So I do have a few more people that yet need to take it, so we won't go over it till hopefully Wednesday. We'll talk about a few questions that were commonly missed, but so overall better. I think one person missed one. I think that was the high score. So again, you can come and look at those exams in my room. In my office, there were 4 of you that skipped the question. So when I say 62 questions, make sure you answer them all. Make sure you're looking over them. So 4 of you skipped one. 2 of you didn't put your test form on, which is not the end of the world because the computer figures out which one works out best. 1 of you didn't bubble your name in, and it was somebody that got an A, so I almost took a point off for that. But I did go back and figure out who that was. Let's see. Yes, I guess that's what I wanted to say. I have gone through all the homework, so if take a second to look back through that, and if there's any that you think, oh, this should have been marked right, why was this marked wrong? Hopefully I didn't miss anything, but at least take a look back at the grade book and see if it looks reasonable to you. But I think I've caught everything. Those fill in the blanks, if you spell it wrong or you bold face it or whatever, it will mark it wrong. So I think I've checked all that. Okay. All right, so where we left off last time, we just started the chapter on innate immunity, chapter 16. And we just kind of started talking about some of the physical barriers, as well as the role of normal microbiota. And we said that there is the relationship that we experience with our mostly bacteria, other organisms as well, that it still seems like it's too loud, that live in our body, mostly in our large intestine, but lots of other places as well. We think that those organisms are really serving us a great service and outcompeting other organisms that might be more pathogenic. It's kind of a mystery as to why our immune system allows some of these microbes to live in our body. Why doesn't it get rid of it? I don't have a great answer to that question, but it does appear that those microbes serve a really important role to keep our immune system stimulated. Because they've done some really cool experiments with mice that are born into sterile conditions and raised in sterile conditions. And they found that those mice don't get very big. They're kind of scrawny looking. And if you do introduce some microbe, they don't have very much of a immune response to it. So things that wouldn't normally even get a mouse sick are likely to kill it. If it doesn't have that normal microbiome. So we think that, again, those bacteria that we live with and other organisms help keep our immune system stimulated and working a lot quicker. And of course, our microbiome improves our overall health in ways that we are just really beginning to understand. We know that it helps that there are bacteria that produce a B vitamin for us. We get a little folic acid from bacteria. Of course, that's necessary to make DNA. We get vitamin K, which is helps in blood clotting. So we're just starting to understand some of the beneficial aspects, and hopefully someday we can understand maybe what probiotic we can take some live bacteria that might make us healthier. Really, I mean, there's a lot of probiotics on the market, but we really have no idea whether they do any good or not. We do have one treatment. Remember, if you take antibiotics and that wipes out, some of your variation of microbes and allows something to overgrow, like C. diff, really, they always give antibiotics first to get rid of C. diff, but if that doesn't work, a procedure that is probably going to take off here eventually is adding back some of that normal bacteria that a person has lost. So we can do that by fecal transplantation. They call it fecal microbiota transplantation, but they take a donor, maybe a family member, and put it in the colon, usually by an endoscope, although they can do it in pill form now too. And that usually does a great job of filling in all those microbes that were lost due to taking antibiotics. It's not perfect. There's been a couple people that have gotten infection from the feces that was transplanted, but once we learn how to screen for that, I think that is going to be a great treatment for C. diff. Okay, so just a little sum up of some of we mostly been talking about some physical barriers. I brought in some kind of chemical defenses at our skin and our mucous membrane. So it's just a nice little kind of wrap up from your OpenStax there as to where we are so far. Some of these mechanical defenses, things like shedding skin cells. We shed cells at our mucous membranes as well, not quite as quickly. Remember, our skin is the outer cells are dead, so then pathogens can't adhere. Okay, so really important. That's section 16.1, kind of physical barriers. 16.2 gets into chemical barriers. So there's a lot to talk about here. We'll start first with a little discussion of complement proteins. We'll get into cytokines on a very general way, and then we'll start getting into some immune cells and phagocytosis and things like that you normally really think about when you're talking about innate immunity. All right, so what is complement? Complement is a series of, there's about 30 different proteins, and these are present in our bloodstream, blood plasma, so the fluid portion of our blood. And these are proteins that were made in the liver. So they're circulating in our blood, and then they'll leave the bloodstream and go out into the tissues when they get during inflammatory responses. So we'll talk about inflammation more and how that works as well. It's considered part of the innate system because these proteins will act the same no matter how many times we've encountered that pathogen. So it's not like a learned response, in other words. And it's considered a cascade reaction. So these proteins are circulating in an inactive form, and then there's several different ways that the cascade can get activated, but one protein will act on another protein that causes that protein to act on another protein. Okay, that's what we mean by cascade reaction. More in detail on how that happens, but just first as kind of an overview, well, what do these complement proteins actually do? They can act as opsonins, and I think that's the term that I brought up already. An opsonin is something that's going to increase the chances that phagocytosis will happen. So opsonization, that's the coating of an invader. So that phagocytes can more easily engulf cells. Okay, so we'll get some examples of that in a minute. Some of these complement protein fragments also act to increase inflammation and chemotaxis. Now, most of us are programmed to think about inflammation as being a bad thing, but in small controlled amounts, it's a good thing, and it's necessary for our immune cells to get to places that they need to be. Okay, so chemotaxis, that means they're going to attract other immune cells. To the area where there's some injury or pathogen present. And also cytolysis by MAC. MAC stands for membrane attack complex. So these first two functions are really, when you think about complement, that means something that's helping something out. Okay, so these first two functions, these are just kind of helping other aspects of immunity. They're not actually killing cells, invaders themselves, these proteins, but this last function, they're actually acting on the pathogen to poke holes in the membrane. That's going to bring about the lysis of that pathogen, and it's certain kinds of pathogens. Okay, so that's just a little overview of how complement, what it actually does, and kind of trying to visualize that overview of a complement pathway. Like I said, there's 3 different ways that these proteins get activated. One of them is when we have some antigen antibody complex. So those are terms that we're going to talk more about, but an antigen is something that elicits an immune response. So something like, say, here's a bacterial cell, some protein on the surface of the bacterial cell acts as an antigen, and an antibody that is part of adaptive immunity, we'll talk more about those later, binds to that antigen, and that acts on the first complement protein, C1. It's actually a protein complex, but that can act to split, to make that C1 complex. I think it does actually split, but it acts on other complement proteins to split them. Some of those bits come together to act on another protein to split it. Okay, and then some of these bits are going to act as opsonins. Some are going to act in chemotactic factors. Some of them will increase inflammation. And then some other ones can come together on the surface of a bacterial cell and actually poke holes in the membrane. Okay, so just kind of, again, a little overview. Your book, your OpenStax book, I'm going to show you the figure that they show, and it's really lame. So I wanted to bring in this other figure, which I know probably looks a little bit more complicated than what you would hope. I'm going to make a note of the things that I really want you to take away from it. So just listen to me as I talk through it, and then I'll highlight what I want you to understand. Let me just change colors there a little bit. Okay, so all right, so here we've got a bacterial cell. There's some antibody that has bound to the antigen on the surface there. Again, that's only one of the ways that complement can be activated. But here's that first complement protein, C1. And when it gets activated, it's acting on the C2 and the C4 proteins. When a complement protein splits, they name the bits that result A and B. Okay, so when C2 splits, it becomes C2A and C2B, and C4 splits and becomes C4A and C4B. Some of those bits come together. In fact, it's C4B and C2A. They come together, and that protein has a special name because it's important. They call that C3 convertase. Okay, and that acts to split protein C3. Okay, so when C3 splits, now we've got some bits that are actually going to do something. The C3A, that triggers inflammation, and we'll get a little bit further how the mechanism of how that actually works briefly. C4b acts as an opsonin. Okay, so that's one thing I don't really like about this diagram, but C3b actually will come up here and bind to the surface of the bacterial cell. So now that's like a handle that pseudopods can grab a hold of and more efficiently phagocytize that bacterial cell. Some of the C3b, though, will bind with this leftover C3 convertase and act on C5 and split C5 into C5a and C5b. Okay, the C5a will trigger inflammation. The C5b comes up here, you can see it up here, binds to the outside of that pathogen along with C6, C7, C8, and lots of C9s to make that membrane attack complex. Okay, so what I want you to remember here, that C3a and the C5a, they act to increase inflammation and chemotaxis of cells. Okay, so that A, think allergy. Okay, when you think allergy, you think inflammation. So the C3A, C5A, those bits will call other cells to the site, other immune cells, and act on some other cells to release inflammatory mediators. And then the B, the C3b and the C5b, they are going to bind to the pathogen. So B for bind. Again, C3b acts as an opsonin, and C5b, along with C6, C7, C8, and C9, form that membrane attack complex. Okay, so I try to be a little bit more clear than usual in the objectives for this. So like what I want you to take away. But this is pretty much it here. Just kind of the big picture of how it's a cascade reaction and what bits are kind of especially important. Here's a transmission, I think it's transmission, maybe it's a, transmission electron micrograph of a cell that has been attacked with membrane attack complexes. So you can see all these little holes. They've gotten its outer membrane full of holes. The membrane attack complex, gram-negative cells are going to be a lot more susceptible to this. Gram-positive cells have just too thick a layer of peptidoglycan, and those complement proteins can't penetrate through there. But the gram-negatives, remember, outer membrane, really thin peptidoglycan, and those membrane attack complexes can penetrate all the way through into the plasma membrane. So anytime a cell is having its plasma membrane disrupted, that's bad for the cell. Okay, so we talked about how soaps and detergents can do that. A lot of antibiotics some antibiotics, polymyxins act on the cell membrane. Okay, but that again, that's mostly gram-negatives. Kind of just backing up to finish up. How are the different ways that complement proteins can be activated? Well, the first one we talked about already, the classical pathway. That's when an antigen antibody complex, oops, got ahead of myself. Will cause C1 to act as an enzyme. And we call it the classical pathway because it was the first one that we described. But actually in the course of an infection, it's not going to be the beginning way that complement gets activated because it takes a while for antibodies to get produced. But so again, we call it classical because we figured it out first. With the alternative pathway, C3, we'll try to simplify this. C3 is split by pathogens. I've got a graphic that I'll show you that in a little bit more detail in a second. And with the lectin pathway, C3 is split by microbial sugars bound to lectin. Okay, so again, let me show you a picture that will hopefully make that more sense. Here's a YouTube video that kind of goes through complement really quickly. But the nice thing is you can kind of watch it a couple times, and it's always nice to hear it from somewhere else besides me. So that's a good video that's just about at the level that we're studying it here. Here's the 3 ways of activation. So the classical pathway involves the antibodies. With the alternative pathway, I said that C3 was split by the pathogen. Well, C3 is actually being split on a low level all the time without any activation. And when it's near a pathogen, some of that C3b will bind to it, and then much more C3 will be split. It involves a couple other protein factors here that have to be around. We're not going to worry about what those are. Okay, so again, when it's in the immediate vicinity of the C3b that's just been split, lots more C3 will get split. And then with the lectin pathway, a lot of bacteria have a sugar on their surface called mannose, and that's a sugar that our cells don't ever have on the surface. So it's something that really tags them as being different than our cells. So found on the surface of many bacterial pathogens, we produce lectins in the liver, and they are proteins that bind to lectins, and then that mannose-lectin complex can act as a C3 convertase to split more C3. Okay, so those are the 3 ways that C3 gets split. So what activates the complement cascade can be different, but you still will get the same results. You'll still get the C3A and the C5A and the bits that act to increase inflammation, the bits that act as opsonins, and the membrane attack complex. So again, however way it's activated, you still get kind of the same result. Okay. And remember, all this activation is taking place in the blood. Those bits will move out into the tissues wherever they're necessary to work. Okay, so this is the figure that they give you in your book. So it's a lot of describing in words in the paragraph, and then there's nothing wrong with this, but it's just kind of hard to visualize, I think. So okay, your book is a fine resource, but again, pay close attention to especially what I've highlighted here in lecture. Okay, questions about that? It's inherently a little bit confusing. Okay, the other, in terms of, a kind of a chemical response, cytokines, we're going to talk about a lot of different cytokines, mostly in a very general way. What are cytokines? They're kind of, we could think about them as messengers, chemical signals that cells send to other cells to be in communication with them. And there's going to be lots of different cytokines, like I said. I just want you to be kind of aware of the different kinds of messengers that are considered cytokines. Usually I'm not going to be worried about which cytokine it is. So for example, there's about 36 different interleukins. Okay, so IL-1, IL-6, IL-12. Okay, we have them numbered that way. So interleukin, that means between white blood cells. They decided a number of years ago that that wasn't a great way to describe these chemical messengers because a lot of these interleukins actually act on cells that are not white blood cells. So they decided to call interleukins a kind of cytokine. But some names are too good to give up, so we stuck with them. A lot of times, again, I'm not going to be concerned which interleukin it is, just that a cytokine is making some change happen. Chemokines, those are cytokines that are acting specifically for chemotaxis of cells, to bring other cells to some spot where there's an infection or injury. Tumor necrosis factor is another cytokine that you may have heard of. We call this cytokine this because when it was discovered in mice, and they added it in larger quantities to mice, they found that it actually got rid of tumors. So they thought, you know, to begin with, oh, we're going to cure cancer, but when we add this to humans, it causes such galloping inflammation that it's enough to kill a person when you add too much of it. And in fact, a lot of autoimmune diseases, the contributing factor is that people produce too much tumor necrosis factor. So there's drugs like Humira, that's one that you may have heard of. It's actually a tumor necrosis factor blocker. So people with like rheumatoid arthritis or Crohn's disease that are on Humira, it's to kind of dampen down some of these cytokines. So again, nothing specific about those. I do want to talk a little bit more about interferons in a minute, but this is just a figure from your book that talks about how these cytokines work. Just to know that sometimes cytokines are working in a way that we would call autocrine, meaning they're actually chemical messengers to themselves, which doesn't seem very efficient, but they secrete the cytokine and then they have receptors for that cytokine. It's like reminding themselves to do something maybe. But some cytokines are self-stimulating. Some cytokines are paracrine, meaning that they act on cells really close by. And others are considered endocrine. So those cytokines get in the bloodstream and are going to affect cells all over the body. And another thing about cytokines that a lot of them have overlapping function. So it makes things a little bit confusing. But there's two different interferons, kinds of interferons. There's type 1 interferon and type 2 interferon. They have very different functions. I want to talk especially about type 1 interferons here, just kind of briefly. That's interferon alpha and interferon beta, because that's one of the innate system's only response to virally infected cells. It's one way to kind of slow down viral infections. And then interferon gamma, which is a type 2 interferon, has a totally different function. It's secreted by different kinds of cells. And because it's secreted by T cells, we consider it part of adaptive immunity. And we're not really going to worry about that very much. So your book doesn't go into too much detail here about interferons. Again, so it talks about the 3 different functions of interferon. This one down here is the gamma interferon that activates immune cells. I'm not going to worry about that. Again, the type 1, the alpha, I'm drawing it sideways. Let's just call it type 1, alpha and beta interferon, primarily are when a virally infected cell is what's going to secrete the type 1 interferons. And that interferon doesn't actually help that cell that's secreting it. Once a cell is infected with the virus, it's pretty much nothing that our immune system can do to clear that other than sacrifice that cell. Okay, so when it secretes interferon, it's to help neighboring cells. So when it acts on, it may have some effect. Your book doesn't really talk about this, even though it mentions it in this figure here. It may have some kind of effect on neighboring cells that are infected. but primarily it has an effect on cells that are not yet infected with some intracellular pathogen like a virus. So what does that do? It actually signals those neighboring cells to make an inactive protein that will come in handy if at some point later the cell is infected by a virus. Okay, so we won't worry about, and it's like usually they call it AVPs, antiviral proteins that are inactive. And then at sometimes later, if this cell does become infected, then it let's see, if it doesn't become infected with the virus, then it will cause the cell to stop synthesizing proteins. Okay, that's what the virus wants to do, take over that cell, and then the virus will start taking over the ribosomes to make its own proteins. So if this inactive protein causes the cell to stop protein synthesis, doesn't that harm the cell? Yes, it does, but it can kind of maintain for 3 or 4 days, usually without making any new proteins, but it's going to slow viral replication. Whoops, got to the edge of my thing here, slow viral replication. So that unfortunately causes some symptoms in the host. How do you feel when you're infected with a virus? Usually you get kind of achy and tired. That's because your cells are secreting this interferon. So it has some effect on our cells as well, but again, it's supposed to slow down the spread of, keep the virus from replicating so quickly so that it gives a chance for our adaptive immunity to mount a more specific response. And then this is just kind of a sum up of the chemical defenses. Before I came in, I thought, oh, maybe I shouldn't have even posted this because I don't need you to learn all this detail. In some cases, you know, it talks about things that we've talked about. And then I think, but there's a lot of other stuff too that we didn't really get into, but there's a lot of chemicals in bodily secretions that are going to act against pathogens that we don't even think about, like, you know, acid in the stomach. Well, that's to digest our food, right? Well, it also has this other function of killing a lot of pathogens that we swallow. Same thing with digestive enzymes. Yeah, it's digesting our food, but it's also killing pathogens. And one function that some of these antimicrobial chemicals have that we see that I'm going to mention again later, so I'll mention it here, is that some of these chemicals are going to sequester available iron. Especially bacterial pathogens, iron is often a limiting factor. So if the bacteria don't have access to iron, they can't grow and reproduce. So some of these chemicals will stimulate kind of the uptake of our cells by any available iron, so it's not available for pathogens. So here they talk about complement a little bit, and then maybe a little bit more detail than what I did. It talks about some different cytokines, what they do, and we're going to get into this pretty soon here, talking about inflammation and some of those inflammatory mediators. But again, probably a little bit more detail than what I meant to go into there. Okay, and then when you think about innate immunity, really the first thing that you think of is the cells that are working. So figure 16.3 here talked about the formed elements of the blood. So that just means the solid parts and not the plasma. So formed elements, typically platelets, red blood cells, and white blood cells. When you think about formed elements. So where do those cells come from? Well, it all starts out with some stem cell in the red bone marrow. Of course, a stem cell is a cell that is capable of differentiating into any kind of cell. And through a process called hematopoiesis. That's just a differentiating, fancy way of saying differentiating. Okay, cells are going to differentiate into different kinds of cells, precursor cells like myeloid stem cells and lymphoid stem cells. And those stem cells then will differentiate into things like platelets. Platelets actually do have some immune function. Mostly they are for clotting blood. That's their main function. So I won't really talk much more about those. And of course, red blood cells or erythrocytes, their main function is to carry oxygen to our tissues. As far as I know, they do not have any immune function. Mast cells, I'm just going to mention them briefly here. We think about them as being important in inflammatory reactions, just to kind of simplify things, though I'm not going to talk about those very much. I do want to spend more time on the basophils, neutrophils, and eosinophils. Those are also known as granulocytes because they have granules in them. We'll talk about those in a little bit more detail. And monocytes in the bloodstream, when they leave the bloodstream, they differentiate into macrophages and dendritic cells. Okay, so those are white blood cells, also called leukocytes, okay, as well as the granulocytes. Those are white blood cells as well. These lymphoid stem cells differentiate into natural killer cells. Sometimes we call them NK cells. We're going to mention them a couple times, but not very much. We are going to spend a lot more time on our lymphocytes, our T cells, and our B cells. The B cells will further differentiate into plasma cells that end up making lots of antibodies. So we'll talk much more about that as we get into our next section in adaptive immunity. Okay, so the macrophages, the dendritic cells, the lymphocytes, those are called agranulocytes because they don't have granules. Okay, I'll just write that here. I have that written again later. Okay, so again, that's the monocytes that become macrophages, dendritic cells, NK cells, T cells, and B cells. Those are all agranulocytes. So we have lots of different names for different kinds of cells. Let's see. All right, so let's talk about some of them in a little bit more depth. I show pictures of them. You don't need to be able to identify them. Take a good histology class if you want to learn how to do that. But when we stain them with little different dyes, then they look a little bit different and we can tell them apart usually. Although apparently it's really hard to tell the difference between B cells and T cells under a light microscope. So the neutrophils. We have a lot of neutrophils. Somewhere between 50 and 70% of our white blood cells are neutrophils. They are also, let's say of total white blood cells. They are also called PMNs. And that stands for polymorphonuclear leukocyte. Okay, so a leukocyte is a white blood cell. So that name, polymorphonuclear, it makes sense when you look at it here because that purple thing in the center of that cell, that's the nucleus. Many-lobed nuclear, polymorphonuclear leukocyte. Now it turns out that eosinophils and basophils also have that lobular nucleus, but because most of the granulocytes are neutrophils, that we just, polymorphonuclear leukocytes are mostly neutrophils. So sometimes we just use that term synonymously, even though it could be used to include the eosinophils and basophils as well. These are highly motile cells. So they are leaving the bloodstream in by amoeboid action, moving out into the tissues where they're necessary to clean up after an accident or fight a pathogen. They're considered first responders. They're usually the first ones there. They're following the chemical signal to where they need to do work. And they are extremely short-lived. Maybe only about 24 hours or less once they leave the bloodstream. So it's not usually considered a bad thing though. So we'll talk about why that's okay that they don't live very long. They get replenished really quickly. How do they act? They do a lot of different things. They act a lot of different ways. They're phagocytic. And we're going to talk more about phagocytosis, but remember that means cells that eat. Okay, so here's a picture. This I just got off of Wikipedia. This is a neutrophil that's phagocytizing some anthrax. It's kind of a cool picture. I don't really understand. What I don't understand about this is that those anthrax bacteria should be way, way, way smaller than that. I mean, this is like a snake python eating an antelope or something here. Like, it seems like the neutrophil should be way, way bigger than that, but I don't know. That's what it said, according to Wikipedia anyway. So neutrophils can act by phagocytosis. They can also act by degranulation. Okay, so these little granules that they have are full of digestive enzymes. Okay, so they can expel digestive enzymes. Enclosed in the granules. Okay, so that's the primary reason that we want them to be short-lived, because if they're degranulating, those digestive enzymes that are in those granules, they're acting on our cells as well. So we don't want them to keep doing it over and over and over again. Okay, degranulate a little bit. We need more. We'll get some more cells to do it some more, so we can kind of dial in a right response. There's one other way that neutrophils can act, and this was just described fairly recently. It's called the formation of nets. Okay, so that stands for neutrophil extracellular traps. And this is, you can kind of think of this as a neutrophil being kind of a kamikaze pilot, okay, that's going to sacrifice itself in order to fight against that pathogen. They actually kind of expunge some of their chromatin, some of their DNA, and it acts, expunge it out of their cells, so they're sacrificing themselves. And this is a great picture here showing it's almost like a Spider-Man net or something on this Klebsiella pneumoniae in a mouse lung. And it's kind of, you can see how it's just immobilizing that bacterial cell so that it can't invade any deeper. And there's some antimicrobial proteins involved with that DNA as well. So again, this is something that only neutrophils can do, and it's just kind of a cool thing. If you've ever extracted DNA from anything, the DNA is kind of sticky, so it's kind of easy to picture how that might work. So this is just a graphic that I got online that just summarizes the three ways that neutrophils can work: phagocytosis, degranulation, and this particular site called it netosis instead of nets, neutrophil extracellular traps. So just kind of visualize that a little bit better. Okay, and then the other kinds of granulocytes are the eosinophils and the basophils. They have a lot of overlapping function, so I just presented them together here. You can see they're much more rare in terms of the total white blood cell count, at least in the bloodstream anyway. Mostly they act by degranulation. Sometimes eosinophils can be phagocytic, and I have read in some places that basophils can be phagocytic. I think your book says they're not, so I put that in there. But they act, again, mostly by secreting these granules. They are both kind of famous for targeting large eukaryotic pathogens like helminth worms. Okay, so they act by degranulating on these larger pathogens that can't be phagocytized because they're much larger. And we also see, like if you do a blood count of a patient that's got some infection going on, if they've got an elevated number of eosinophils, then that's kind of a dead giveaway that either the patient is dealing with some eukaryotic pathogen or the patient's having an allergic response. Okay, so we see more eosinophils produced in allergic responses as well as fighting helminth worms. I came across this picture. This was on my Pinterest feed. So I don't know what algorithms those people are doing at Pinterest, but they have got me pegged pretty well. So I don't have this cited. I don't know. I couldn't figure out where it came from. But here's a fluke, kind of immature stage of a fluke worm. And you can see all these eosinophils mobbing it, degranulating it. You can see how it would take a lot of eosinophils to have any effect on a big pathogen like that. Okay, and then we'll just kind of briefly mention some of these lymphocytes here, because the B and T cells are most active in adaptive immunity, but they are a pretty good proportion of our total white blood cells, but they mostly don't circulate in the blood. They're mostly going to be found in the lymph. And we'll talk again more about that later. Just kind of briefly here, what do B cells do? They are going to differentiate into plasma cells, which then are going to make antibody. So we call that kind of response when B cells get activated, we call that our humoral immunity. And that's kind of an older term that maybe you don't hear anymore, but the body's humors are sort of like the liquid things that we have in our body, like the blood. Okay, so B cells are acting on pathogens that are in our bloodstream. So really active against bacterial pathogens or maybe toxins produced by bacteria. And then T cells, T helper cells, and cytotoxic T cells. Are the two different kind of most famous kinds of T cells, and they are going to function in cell-mediated immunity. So mostly going to be helpful against viruses. Okay, cells acting on our own cells to get rid of that viral infection. There's the third kind of lymphocyte. Again, I'm not going to mention these a whole lot. Natural killer cells. They are named that way because at first we didn't understand how they killed other cells. That they just naturally do it. We didn't understand the signaling that was involved to get them to act on other cells. We now know that they are primarily acting on cells that are not normal in some way. So in this graphic in your book, this gets ahead of ourselves a little bit because we're going to talk about these MHC proteins later, but all cells have a way to advertise what's going on inside their cells. And if cells are not advertising that, that's when the NK cells are going to act on them to kill them, if they're not doing their normal advertising of what's going on inside them. So we'll get into more about that later. All right, so we'll pick up next time talking about macrophages and dendritic cells and get into inflammation and probably start our adaptive immunity. Have a good day.